Hi, everyone. This is Kara Burke, a pharmacist in the Division of Drug Information. I would like to welcome you to this educational activity sponsored by the FDA Center for Drug Evaluation and Research, Office of Communications, Division of Drug Information. Today's webinar is titled FDA Drug Topics, Development and U.S. Regulation of Preventative Vaccines. Before I introduce our speaker, I have a few housekeeping remarks. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. Unapproved use disclosure. CE faculty, speakers, are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. Disclosure. The faculty's planning committee members and the FDA CE consultation and accreditation team have nothing to disclose. All inquiries for information relating to our webinars should be directed to ddiwebinars@fda.hhs.gov. We hope you will enjoy meeting our presenter today. Captain Julianne Valancourt is a pharmacy officer in the United States Public Health Service Commission Corps. In 1995, after practicing hospital and community pharmacy, she joined the United States Public Health Service Commission Corps, starting at the FDA, first as a regulatory project manager and then as a team leader in the Office of Vaccines Research and Review in the Center for Biologics Evaluation and Research, or CBER. There, she contributed to the review and licensure of vaccines that included the first two pneumococcal conjugate vaccines and the first human papilloma vaccine. In 1999, as a United States Public Health Service Commission Corps officer, she served in the Office of Foreign Disaster Assistance at the U.S. Agency for International Development. She returned to the FDA in 2001 and continued to work in the Office of Vaccines Research and Review. Since 2015, she has served as a policy advisor and rare disease liaison in CBER's Office of the Director, where she coordinates CBER's rare disease program. Now, please give a warm online welcome to Captain Valancourt.
Thank you so much, Kara, for that kind introduction. It's really a pleasure to be here today to be able to give this presentation on the development and U.S. regulation of preventive vaccines. And before doing so, I'd like to thank the organizers and my colleagues in the Division of Drug Information and the Center for Drug Evaluation and Research at FDA. And I would also like to thank and acknowledge the Office of Vaccines Research and Review and the policy staff in the Center for Biologics Evaluation and Research for their support. So here's my disclosure statement. My comments are an informal communication and represent my own best judgment. These comments do not bind or obligate FDA. As you can see from the objectives listed on this slide, my talk will be a high-level overview on how and why FDA regulates preventive vaccines. I will not be focusing on specific vaccines, but rather plan to provide you with the historical basis for vaccine regulations so that you can appreciate why we regulate them the way we do. I will also provide an overview of the vaccine development process, and that will be mostly from a clinical perspective. So let's start by looking at the legal framework within which FDA operates. Now, this may seem like an elementary review, but I think it will be helpful to set the stage for my talk. So the United States Constitution is the fundamental source of authority for federal law. Legislation is enacted by Congress in the exercise of its legislative or lawmaking authority. Congress can delegate some of its lawmaking authority to regulatory agencies like the FDA. FDA can then use that authority to promulgate regulations which, like statutes, have the force and effect of binding law. FDA can also issue guidance documents which are not legally binding. These documents can be very useful for regulated entities because they let stakeholders know the FDA's perspective on certain topics and issues that are typically not covered in adequate detail by regulations. So let's look at a few vaccine-specific guidance documents as examples. So here on this slide, I just have the titles of a few vaccine-specific guidance documents. So you can see there's one on considerations for developmental toxicity studies for preventive and therapeutic vaccines for infectious disease indications. Another on the clinical data needed to support the licensure of seasonal inactivated influenza vaccines. That latter one was from May of 2007. The first one I mentioned is from February. I'm trying to read it myself. I believe it's 2008 or 9. And then you can see some more recent ones that concern development of COVID-19 vaccines. So there's the development and licensure of vaccines to prevent COVID-19 that was issued in June of 2020, and one on the emergency use authorization for vaccines to prevent COVID-19 that was issued in 2021. And here on this slide, I show some examples of general topics guidance. So here's one on formal meetings between the agency and sponsors of applicants of PDUFA products, another on investigator responsibilities, and a third example on demonstrating substantial evidence of effectiveness for human drug and biological products. So these are just a sampling of guidance documents, and there are many, many more, and you can find them by visiting fda.gov and searching on guidance documents. So FDA's regulation of biologics is authorized by two main substantive statutes. One is the Federal Food, Drug, and Cosmetic Act, or the FD&C Act. The FD&C Act is relevant to all the products that FDA regulates, including biological products, and it has been amended over 100 times. The other important statute is the Public Health Service Act, or the PHS Act. The PHS Act includes two sections that are key to FDA's regulation of biological products, including vaccines. One is the licensing provisions, which give the FDA the authority to license biological products, and Congress has amended this section about 11 times. And the other is the communicable disease provisions, which gives FDA the authority to issue regulations to prevent the spread of communicable diseases, and this section has been amended about 4 times. So we're now going to look at the history of these laws and some important amendments to them. So in the 1890s, there were several biologics in worldwide use. These included live attenuated vaccines against smallpox and rabies, heat-killed vaccines against cholera, typhoid, and plague, and antitoxins against diphtheria and tetanus. These antitoxins were referred to as serum therapy and were an application of passive immunity. These products were manufactured by injecting a small dose of diphtheria or tetanus toxin into animals, horses for the most part, to produce a serum containing antitoxins or antibodies against the toxin. Large-scale production of antitoxins started in Europe and soon spread to the United States, again at the end of the 1800s. And these vaccines and antitoxins had a dramatic effect on the rates of disease and deaths due to the causative agents. By 1895, several countries in Europe, including France, Germany, Italy, and Russia, had enacted laws to regulate such products. In the early 1900s, diphtheria patients were routinely treated with antitoxin derived from the blood serum of horses. In 1901, 13 children in St. Louis died after receiving diphtheria antitoxin contaminated with tetanus spores. The serum had been manufactured in local establishments with no central or uniform controls in place to ensure potency and purity. There were also no inspections or testing of the final product. Around the same time, a similar event occurred in New Jersey, where 9 children died from tetanus after receiving contaminated smallpox vaccine. These tragedies spurred Congress into passing the 1902 Biologics Control Act, also called the Virus Toxin Law. So in addition to requiring for the first time pre-market approval of certain biological products and authorizing the inspection of their manufacturing facilities, This legislation included product labeling requirements and provided for penalties for violation of the Act. The Biologics Control Act was not only the beginning of federal regulation of biological products, but actually it was the beginning of federal regulation of any medical products. In 1905, there were two publications that were significant for the oversight of products now regulated by the FDA. Samuel Hopkins Adams published The Great American Fraud, which was comprised of a series of articles exposing the false cure-all claims made about patent medicines and revealing that in some cases, these products were damaging to the health of people using them. The articles provoked public outrage over these worthless and dangerous products. In addition, Upton Sinclair published The Jungle, a book in which were described the practices of the meatpacking industry in the contaminated and tainted meat the industry produced. Again, the public was outraged. This public sentiment spurred Congress to act. In the following year, Congress enacted a federal statute for the regulation of food and drugs. Passage of the Federal Food and Drugs Act, also known as the Pure Food and Drugs Act, in 1906, was the beginning of federal regulation of food and drugs. Prior to that, the individual states took it upon themselves to certify the quality and purity of food and drugs sold within each state. This law focused primarily on enforcement. The Federal Food and Drugs Act prohibited the misbranding and adulteration of products and provided for criminal penalties, including imprisonment and fines, for violation of the statute. In addition, it authorized product seizures. A seizure is a court action against a product, not a person, through which a court can require that the product be held in place and not moved during legal action by the government. However, the Federal Food and Drugs Act did not require pre-market approval of drug safety, and this had this was basically an oversight with deadly consequences. In the 1930s, the drug sulfanilamide had been used to treat streptococcal infection safely and effectively for some time. It had been used primarily in tablet or powder form, but in 1937, a salesman reported demand for the drug in liquid form. The manufacturer's chief chemist experimented, and he found what was thought to be a suitable solvent. The company then distributed 633 shipments of the mixture all over the country. Within 2 months, more than 100 people in 15 states had died. Before marketing the product, the manufacturer's lab had tested it. However, it was tested for flavor, appearance, and fragrance, and it was found to be satisfactory on those attributes. However, the new formulation had not been tested for toxicity. At the time, the law did not require that safety studies be done on new drugs. Because no pharmacological studies had been done on this new preparation, the chemist failed to note one characteristic of the product. The solvent he used, which is diethylene glycol, normally used as antifreeze, is toxic. Sulfanilamide elixir, which contained an unlabeled toxic solvent, and the tragic deaths that resulted from the products used prompted Congress to take action again. Whoops. In 1938, Congress enacted a landmark piece of legislation, the Federal Food, Drug, and Cosmetic Act, or the FD&C Act, which significantly increased FDA's authority to regulate drugs. The new law required that the manufacturers provide evidence of safety before they could market a drug product. It added a new regulatory tool, injunctions, which are court actions ordering a firm to do certain things, such as shut down its production of a product. The FD&C Act also extended regulatory authority to products beyond drugs, such as cosmetics and therapeutic devices. The 1938 Food, Drug, and Cosmetic Act did not, however, require evidence of efficacy before a medical product could be marketed. Now we're going to switch gears a little bit. In 1944, which was 6 years later, Congress compiled a number of statutory provisions that existed in various laws into the Public Health Service Act, which is a key statute for FDA Center for Biologics Evaluation and Research, or CBER. And this Act's provisions included the requirement that biologics be licensed and provided authorities related to the control of communicable diseases, as I previously noted. Then moving along chronologically, in the late 1950s and early 1960s, tragedy struck again. Starting in 1957, the drug thalidomide was widely sold in other countries and prescribed to pregnant women to treat morning sickness. In 1960, the company applied to FDA for approval, but a medical reviewer who had just started working at the FDA, Dr. Frances Kelsey, blocked its approval because of her concerns about its safety. Indeed, by 1961, it was obvious that thalidomide had caused serious birth defects in thousands of children. So again, Congress was spurred to take action. In 1962, in response to the thalidomide tragedy, Congress passed the Kefauver-Harris Drug Amendments to the Food, Drug, and Cosmetic Act. This significant legislation required manufacturers to prove the efficacy of a drug before it could be marketed. It also put into place many other important new requirements. Also in 1961, Dr. Kelsey received an award from President Kennedy for her scientific judgment and her determination in the fact of pressure from the manufacturer to keep thalidomide off the U.S. market. Years later, FDA created an award, the Dr. Frances O. Kelsey Award for Excellence and Courage in Protecting Public Health, which was first conferred to her in 2010. So now that we've had a quick look at some of the history of FDA's regulation of biologics, let's take a closer look at the Public Health Service Act, which both defines biological products and gives FDA the authority to license such products. Section 351I1 of the PHS Act provides the definition of a biological product, as recently amended under the Biologics Price, Competition, and Innovation Act, or PCI Act of 2009, and also under the Further Consolidated Appropriations Act, or FCA Act of 2020. So this current definition is provided on the slide here, and I've divided the two components as two separate bullets just to make it easier to emphasize that there are two parts to the definition. So we can read that a biological product is defined as a virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, allergenic product, protein, or analogous product, or arsphenamine, or derivative of arsphenamine, or any other trivalent organic arsenic compound. The second component being that a biological product in its definition is applicable to the prevention, treatment, or cure of a disease or condition of human beings. So as you can see, a biological product must be a specific type of product as indicated in the first bullet and be applicable to preventing, treating, or curing diseases or conditions in people, human beings, and not animals. It's specific to humans. So as a note, on February 21st, 2020, FDA issued a final rule in the Federal Register to amend its definition of a biological product in 21 CFR 600-3H to conform with the recently amended statutory definition and to provide its interpretation of the statutory term protein under 600-3H6. I'm not going to go over that interpretation, but I just want to point it out because there was some changes, and this affects particularly biological products regulated by the Center for Drug Evaluation and Research. And I'd like to point out that in the definition of a biological product, the term virus is defined differently than what we typically consider a virus to be today. And this definition of how it's used here in the PHS Act can be found in 21 CFR 600-3H1 under definitions. And I'm going to read it to you. A virus is interpreted to be a product containing the minute living cause of an infectious disease and includes but is not limited to filterable viruses, bacteria, rickettsia, fungi, and protozoa. So that's an important distinction to know, because when you see it, you might be thinking virus as we define it today, but it has a specific meaning in the PHS Act. So Section 351 of the PHS Act also provides FDA with the authority to license biological products that are distributed in interstate commerce. In order to be licensed, a biological product must be shown to meet the standards of safety, purity, and potency. Manufacturing facilities have to meet appropriate standards, and manufacturers have to agree to allow their facility to be inspected. Licensure is a powerful regulatory tool. If a biological product is in violation of the statute and/or FDA's regulations, FDA has the power to suspend or revoke the license. FDA can take this action administratively without having to take the license holder to court. FDA also has the power to recall products that are no longer safe, pure, and potent. Thus, vaccines are biologics or biological products, and a biological product also meets the definition of a drug. Therefore, a biological product is subject not only to the requirements of the PHS Act, but also to the requirements, prohibitions, and sanctions of the relevant provisions of the Food, Drug, and Cosmetic Act. So now I want to talk a little bit about vaccine licensure. The biologics licensing regulations are found in 21 CFR 601. That is Title 21 of the Code of Federal Regulations in Part 601. And again, to emphasize, licensed biologics must be safe, pure, and potent, and manufactured consistently according to current good manufacturing practices, or CGMPs, to ensure continued safety and effectiveness. I mean, this is a brief synopsis of what you'll find there in those regs, but that is really the gist of what is required for licensure. So let's look at the definitions in the biologics regs under the definitions section, which is 600.3. I want to look at the definitions of safety, purity, and potency, because they're so critical to the regulation of biologics. So here, this is directly taken from the regulations. So the word safety means the relative freedom from harmful effect to persons affected directly or indirectly by a product when prudently administered, taking into consideration the character of the product in relation to the condition of the recipient at the time. And then the definition of purity in the biologics regs means relative freedom from extraneous matter in the finished product, whether or not harmful to the recipient or deleterious to the product. Purity also includes, but is not limited to, relative freedom from residual moisture or other volatile substances and pyrogenic substances. The word potency is interpreted to mean the specific ability or capacity of the product is indicated by appropriate laboratory tests or by adequately controlled clinical data obtained through the administration of the product in the manner intended to effect a given result. And potency, thus, encompasses the effectiveness of a biological product. So now let's look at the investigational new drug application regulations, or the IND regulations, which are found in Title 21 of the Code of Federal Regulations in Part 312. The Food, Drug, and Cosmetic Act prohibits the introduction of unapproved drugs into interstate commerce. And an investigational new drug application, or an IND, is a request for exemption from the statute, and it's based in the CFR, or the Code of Federal Regulations, and again, under 21 CFR 312. The primary function of an investigational new drug application is to protect human research subjects, and an IND is required for clinical investigations of unapproved drugs in the U.S. So if you go to 21 CFR 312.23, you will find the required contents of an IND. And here it's listed. There's more detail, of course, in the regulations. But you can see there's a cover sheet, which would be the Form 1571, which is available online and fillable, and various other items like an introductory statement, an investigator brochure, which there is a definition in the regs as well, one or more protocols, including investigator information, chemistry, manufacturing, and controls data, or we often refer to as CMC data, pharmacology and toxicology information, including non-clinical data such as animal studies and any previous human experience, if available, on the investigational drug. And you can see that the information that must be submitted, let me go back to that slide. You can basically break it up into three broad areas of information: manufacturing and product, non-clinical data, and then clinical information. So FDA's primary objectives in reviewing an IND are in all phases of the investigation to assure the safety and rights of subjects, and in phases 2 and 3 to help assure that the quality of the scientific evaluation of drugs is adequate to permit an evaluation of the drug's effectiveness and safety. So this is a really important concept and thing to know about what we consider of primary importance when we review an IND. And again, it's safety comes first in all phases, whether it's phase 1, 2, or 3. And then in phase 2 and 3, we also, in addition to evaluating and focusing on safety as a primary objective, look at the design of the study to make sure it's adequate to meet its stated objectives. And this can be found in 21 CFR 312.22. So an IND application may go into effect 30 days after FDA receives the application, unless, based on our review, we notify the sponsor of the IND that the proposed study in the IND application is subject to a clinical hold. And the regulations also include grounds for clinical hold. Now, we may notify a sponsor that the proposed study or studies may be initiated sooner than 30 days if we've reviewed the application within that time and didn't identify any deficiencies that would be a basis for clinical hold. Now, the biologics regulations are found in Part 600 to 681 of Title 21 of the CFR. And as I previously mentioned, the biologics licensing regulations are found in 21 CFR 601. And per 21 CFR 601(2), a BLA is a request for permission to introduce or deliver for introduction a biological product into interstate commerce. The BLA regs require data from non-clinical and clinical studies demonstrating that the manufactured product meets prescribed requirements of safety, purity, and potency, and more, which you will find in the regulation. But the key focus is on safety, purity, and potency. The standard BLA review clock is 12 months, and for priority review is 8 months from date of receipt of the application. So now let's look at the overall basic pathway of vaccine development from the preclinical phase to the post-marketing phase. During preclinical development, in vitro and animal studies are performed to develop the vaccine and test the hypothesis that it will elicit a biological effect in animals. FDA encourages vaccine developers to request a pre-IND meeting with CBER before submitting an IND. A pre-IND meeting provides an opportunity for the vaccine developer to ask questions about the planned clinical development program and product development and receive non-binding advice from FDA. And this in turn might help ensure that the IND is allowed to proceed after the initial 30-day review without a clinical hold. There's no required amount of time after which a sponsor must submit an IND after having had a pre-IND meeting. Phase 1 safety studies typically have 20 to 30 subjects. Phase 2 studies may have around 100 subjects, give or take, And phase 3 vaccine efficacy trials, which again also look at safety, can range from the thousands to tens of thousands of subjects. And the size may depend on the case definition and the primary clinical endpoint and the epidemiology of the disease and other factors. All these considerations go into the design of the phase 3 trials, which are designed to evaluate safety and effectiveness in the target population. Post-marketing studies can be very large, in the high tens of thousands or more, and they're designed to evaluate safety, and sometimes post-marketing studies evaluate effectiveness in specific populations. The original IND submission should provide all the information shown on the slide I previously showed you and talked about, and this would include the protocol for a phase 1 safety study. Beside evaluating safety as the primary aim of the phase 1 study, dose exploration may be done, and this would involve the study having different arms, one for each dose to be evaluated. A phase 1 vaccine study may also be designed to evaluate the vaccine with and without an adjuvant. And often in vaccine clinical development, immunogenicity may be assessed in a phase 1 study. It's trying to look at what type of an immune response is elicited and to measure that in an early exploratory manner. But that's not required. If a phase 1 study has been completed under an IND and the results are favorable, the sponsor typically will submit an amendment to the IND with the results and a protocol for a phase 2 study to evaluate safety, dose selection, and preliminary effectiveness. And I will say that sometimes, frequently, there may be more than one phase 2 study. Establishing a dosing regimen is an important part of phase 2 in vaccine development. Also, evaluating preliminary effectiveness is needed to help design the phase 3 study that will be statistically powered to evaluate effectiveness. A lot of work gets done in phase 2 to prepare for phase 3. FDA recommends that a sponsor request an end of phase 2 meeting to discuss the phase 2 data in the proposed phase 3 study, provided the sponsor plans to continue developing the vaccine product based on the phase 2 data and other issues like manufacturing, which is a whole other discussion that I'm not focusing on in this presentation. An end of phase 2 meeting is not required, but strongly recommended. And FDA has guidance for sponsors on how to request such a formal meeting, and I showed you the title of this guidance document in an earlier slide when I was discussing guidance documents in general. And planning on the sponsor's part is really important for all formal meetings such as pre-IND, end of phase 2, and pre-BLA. There's a required timeline for both FDA in terms of granting and holding the meeting and providing a meeting summary afterward. And there's also a timeline for the sponsor in terms of submitting a background package for review by the review team. And if they don't submit it by the due date for the background package, FDA can cancel the meeting. A phase 3 vaccine and safety efficacy study could take years to complete. In the case of vaccine field trials, when the primary endpoint is clinical disease, the size and duration of the study would be based on the incidence of the infectious disease in the target population over time. This means enrolling enough people in the vaccine and control arms and following them for occurrence of disease, and then comparing the number of cases in the vaccine group to the number of cases in the control group. And the control group might receive a placebo or sometimes an active control, such as a licensed vaccine for a different disease that the subjects might receive anyway, could be used. Overall, the design and plan of the clinical development program for a vaccine product is done on a case-by-case basis. It involves a lot of communication between FDA and the sponsor that should start early on. As preliminary results of the phase 3 trial or trials become available, the sponsor typically will be making plans to meet with CBER in a pre-BLA meeting, which is also not required but strongly recommended. And during this important meeting, the sponsor will present plans for what they intend to provide in the BLA. They'll provide a summary of the clinical data and other data to date, discuss the projected timeline for submission of the BLA, among many other things. The BLA must include a pharmacovigilance plan for post-marketing studies. This plan is reviewed and discussed with FDA during the BLA review. After approval of the vaccine, post-marketing studies are initiated, and they may take several years and will involve very large numbers of people who would receive the vaccine as a marketed vaccine. So here's another graphic that focuses on the three key meeting types that I noted: pre-IND, end of phase 2, and pre-BLA. And the types of information that are typically discussed at each of these three critical time points in vaccine development are noted on this slide. I mentioned some of them. Again, these meetings are not required, but strongly recommended. So there's a certain type of meeting that I didn't discuss on the previous slides that included graphics. This is the advisory committee meeting. So the Vaccines and Related Biologics Advisory Committee, or what we call VRBPAC, is one of the many FDA advisory committees, and there are many. If you watched any of the VRBPAC meetings that were held during the COVID-19 vaccine development process to date, you saw this process firsthand. And transcripts and recordings of advisory committee meetings are available on FDA's public website, fda.gov, and I would encourage you to visit some of them if you're interested. So the VRBPAC is composed of experts external to FDA that are convened upon FDA's request. The VRBPAC is responsible for evaluating data and advising the agency on the safety, effectiveness, and use of vaccines and related biological products prior to certain regulatory decisions such as licensure or emergency use authorization of new vaccines. And the VRBPAC can be convened to advise FDA on more general topics as well. For example, every year there's a VRBPAC meeting held to discuss the strain selection for flu vaccine for the coming season. And recommendations by the VRBPAC are not binding, but FDA usually follows them, but they're not binding. So I want to just revisit this slide before I go into a little bit more detail on safety. I read this before, and it's FDA's regulatory definition of safety found in 21 CFR 600.3. I'm not going to read it, but just want to revisit it to remind you what safety means in a regulatory sense. For vaccine clinical trials, FDA's approach is to ensure that safety is monitored in a layered manner. First, certain solicited events are collected from all subjects for at least 7 days and up to as many as 14 days after each vaccination dose. So if the investigational vaccine has a 3-dose regimen, this would be done after each individual dose. These solicited events include both local and systemic reactions, and subjects may be asked to complete diary cards or use an electronic app to record these events during the study. In addition, unsolicited events, this is the middle box, that's orange, are collected for up to 21 to 28 days after each study vaccination dose. And then in addition, subjects are followed for at least 6 months after completion of all study vaccination doses for any serious and medically attended events. And these different types of safety data from the vaccine efficacy trial or trials are provided in the package insert or labeling for a licensed vaccine. So you can look at the package insert for any licensed vaccine and you'll see the safety data from the efficacy trial presented in this way. Typically, there are tables for the solicited adverse events broken down by local and systemic and broken down by after each dose. And also, you can see the comparison in the rate between the vaccine recipients and the placebo or other control recipients. And then the unsolicited events are usually not in a table, but they may be, but usually there's language about that summarizing the results. And then for the serious and other medically attended events up to 6 months. That's usually also written in a section. So I would encourage you to look at the safety data so you can have an appreciation. There's safety, again, is monitored in these different ways. So we have a comprehensive profile of the safety of a particular vaccine product. So during vaccine development, a big question that comes up is, what would be an adequate safety database for this vaccine in the target population? A safety database is defined as the number of subjects vaccinated with the final formulation, dose, and regimen. And there are many considerations that go into determining an adequate safety database for a given vaccine in development, as listed on this slide. So you can see, you look at the characteristics of the vaccine, early phase safety data, any true safety signals that have occurred to date or theoretical safety signals, the target population, the seriousness of the disease. And for preventive vaccines, we have a rule of thumb for a size of a safety database that's usually considered adequate, at least for in the case of field trials especially. Usually we would look at a safety database of at least 3,000 subjects followed for at least 6 months after completion of the vaccination regimen. And this database size would provide adequate power to detect events that occur at a rate of 1 in 1,000 subjects or higher. So to support licensure of a vaccine, FDA expects substantial evidence of effectiveness from adequate and well-controlled studies, as it does for any drug that would be approved. And I did show you the title of a guidance document in one of my earlier slides that gives advice and guidance in evaluating vaccine effectiveness, and, or not just vaccine, but drug effectiveness in this regard. And to demonstrate vaccine effectiveness, the gold standard is the randomized control trial, or RCT. And an RCT mitigates risk of bias by balancing potentially confounding factors across treatment groups. And typically, a randomized control trial is double-blinded, and it's done in order to further mitigate the risk of bias. For vaccines, a single large multi-center, multi-regional trial is typically conducted to provide sufficient evidence of effectiveness. And we a lot of times refer to such a trial as the pivotal trial for licensure, but I want to point out that the term pivotal trial is not a regulatory term, even though we do use it. So as I stated earlier in the talk, licensed vaccines must be safe, effective, and manufactured consistently to ensure continued safety and effectiveness. There are 3 licensure pathways available for preventive vaccines. Each has a different approach for demonstrating effectiveness. However, each has the same requirements for demonstrating safety and manufacturing consistency. And these approaches are traditional approval, accelerated approval, and animal rule approval. So, I'm getting close to my time. Better speed up. So traditional approval of a vaccine is when the primary endpoint directly demonstrates clinical benefit. And this can be done in different ways. The classic approach is a vaccine field trial, which I mentioned when I showed you that graphic slide of the development process. An example of vaccines that demonstrated effectiveness via a vaccine field trial are the rotavirus vaccines and the COVID-19 vaccines. Another approach is a challenge trial, which is designed to evaluate protection against infection under controlled conditions. And the cholera vaccine is an example of one where a human challenge trial was used to support effectiveness or demonstrate effectiveness. And then another approach is an immunogenicity trial or study, which is when the study is designed such that the immune response is measured. And usually this wouldn't be done for a brand new vaccine, but it might be done for a new vaccine to compare it to something that's existing against the same pathogen. And in this case, you're evaluating the impact of an intermediate clinical endpoint that's been scientifically established to predict protection against the disease of interest. And examples where immunogenicity trials were done are the hepatitis B vaccines when anti-hepatitis B surface antigen IgG concentration was evaluated, and the human papilloma vaccines when prevention of precancerous lesions was used as an intermediate clinical endpoint. So accelerated approval is another pathway. In accelerated approval, a surrogate endpoint that is reasonably likely to predict clinical benefit is used to evaluate vaccine effectiveness. And to be eligible for accelerated approval, the drug, or in this case, the vaccine, must be intended to treat a serious condition and provide a meaningful advantage over available therapies. And it's not mentioned on this slide, but under accelerated approval, there is a requirement that a post-marketing confirmatory study be conducted. An example of a vaccine approved using under accelerated approval is the pneumococcal conjugate vaccine for ammonia in the elderly using opsonophagocytic antibody response as the surrogate endpoint. And then the third pathway is the animal rule pathway. And this requires that the product is being developed to ameliorate or prevent serious or life-threatening conditions caused by exposure to lethal or permanently disabling biological, chemical, radiological, or nuclear toxic substances. And this pathway may be used under certain circumstances, such as the product can't be approved based on other efficacy standards like the traditional or accelerated approval pathways. Adequate and well-controlled field trials are not feasible for one reason or another, and human efficacy trials are not ethical. So the regulations require 4 specific criteria be met. I don't provide those on this slide. I'm not going into detail, but a key point for the animal rule is that post-licensure confirmatory studies be required as with accelerated approval. And an example of a vaccine approved under the animal rule is post-exposure prophylaxis for the use of the anthrax vaccine. And of course, given that we are hopefully on the tail end of the COVID-19 pandemic, I just want to mention emergency use authorization. It, of course, is not an approval pathway, but it is an important pathway to allow the use of a medical product such as a vaccine during a public health emergency. It was established under Section 564 of the Food, Drug, and Cosmetic Act, and it allows FDA to authorize unapproved medical products or unapproved uses of approved medical products to address public health emergencies related to biological, chemical, radiological, or nuclear agents. And certain criteria must be met. EUA declaration must be made, and the the causative agent must cause serious or life-threatening disease or condition. The product may be effective to prevent, diagnose, or treat the serious or life-threatening condition caused by the agent. Importantly, the known and potential benefits of the product must be determined to outweigh the known and potential risks, and no adequate, approved, and available alternative to the product is available. And in this case, it requires prior determination of a threat and declaration of circumstances justifying its need by the Secretary of Homeland Security, Defense, or Health and Human Services. I do want to point out there's a lot of information about the EUA process on FDA's website, and I would encourage you to visit it. So in summary, vaccines are biological products and drug products. The Food, Drug, and Cosmetic Act and the Public Health Service Act authorize FDA to regulate vaccines. The regulations for vaccine development and licensure are found in the 300s and 600s of Title 21 of the Code of Federal Regulations, or the CFR, respectively. The development and licensure pathway of each preventive vaccine follows a basic course from preclinical to post-licensure, each with unique features throughout that are based on several factors in consideration. I have references here, but I think we need to get to the challenge questions. So I encourage you to look at the references, especially the historical ones. And FDA has a great history office. So if you ever come to White Oak, I would urge you to go visit our history office and talk to our historians.
All right. The first one that came in, how does FDA use adverse event information concerning vaccines reported by medical professionals?
Okay, I didn't really talk about VAERS, but VAERS is the Vaccine Adverse Event Reporting System, and anybody can submit a report on an adverse event using a VAERS form, which is a downloadable PDF form on FDA's website. And VAERS is a reporting system that's co-sponsored by CDC and the FDA. And FDA monitors the VAERS data along with CDC that comes in following administration of vaccines, and it's used to detect safety signals that can be further investigated and analyzed, and using various methods and other systems. If there is a safety signal, another system like the Vaccine Safety Data Link or VSD is often used to further investigate it and maybe do a study to see if there's some type of a likely association or not. And that can be, there's information about VAERS. And if there is an association that looks, or if, if further investigation leans towards maybe there being a likely association, typically what will happen is that there's a change in the labeling, and FDA can require a change, or a sponsor may submit a labeling supplement or a prior approval supplement, I should say, to change the labeling to include adverse event information about an event that has been determined after, such a series of post-marketing evaluation, starting with VAERS and um, using other mechanisms afterwards to further evaluate it. I hope that helps. Next one, is there another?
Yeah, we'll try to do squeeze in one or two more. Are there any biosimilar vaccines?
Okay, that's a good question. At this time, there are no biosimilar vaccines. I will say there are biosimilar biological products that are evaluated by the Center for Drug Evaluation and Research, but those products tend to be less complex than the biological products that we regulate in CBER. And so therefore, it's easier or less challenging to demonstrate biosimilarity. But again, to answer the question, at this time, there are no biosimilar vaccines.
Thank you so much, Julie. Okay, let's do, how about one more? What about cancer vaccines or immunotherapy vaccines? Does the Office of
Oh,
Vaccines at FDA regulate those as well? And do they follow the same regulations? That's a good one.
that's an interesting question. Thank you. Cancer vaccines or cancer immunotherapy vaccines are regulated by the Office of Tissues and Advanced Therapies in FDA Center for Biologics Evaluation and Research, or CBER, not by the Office of Vaccines. However, the same statutes and regulations apply, but the benefit and risk is looked at a bit differently, because in the case of cancer vaccines, they would be given to an unhealthy, or a population with a diagnosis of cancer, whereas preventive vaccines are given to a large population of healthy individuals. So the risk benefit ratio is looked at a bit differently, and there are other considerations that are different in clinical development as well.
Okay, thank you, Julie. And I think our time is up. If you had questions that we didn't have time to address today, please email them to our Division of Drug Information email address at druginfo@fda.hhs.gov for assistance. And if to stay connected with the Division of Drug Information, you can scan the QR codes on the slide in front of you to subscribe to our listserv, and also follow us on Twitter. The FDA appreciates your participation in this webinar, and we hope that you will join us again in the future. Our next webinar will be held on November 1st, and the topic will be Understanding Generic Neurotherapeutic Index Drugs. And if you need more CEs for your license renewals this year, please check out our list of home study CE webinars at www.fda.gov/DDI webinars. I think we have about 21 home study CE webinars listed on that page. So be sure to check them out. You may also find additional CE credits, such as FDA's drug regulation course titled FDA's Role in Public Health, drug efficacy, safety, quality, and beyond on our CDER Learn page by entering CDER Learn in the search engine on fda.gov. Thank you again for attending, and please stay safe and healthy. Thank you all.